building blocks of living or non-living things are attracted by that physical forces. This physical force and mechanics are paramount importance for their shape as well as that biological outcome. So far in the biological area, all the biological outcomes are explained in terms of reaction diffusion mechanism, but in some cases direct mechanical force is involved to move the molecule or cells for the complex biological outcome. So, mechanotransduction is a process which converts that mechanical force to all the biological event and that convert the mechanical force to chemical signals by which the cells can do all of its manifestation like say growth, maintenance, differentiation, functioning and last of all that apoptosis. So, in this lecture I shall concentrate on the what are the forces, mechanical forces, particularly say fluid flow and other interstitial flow, and how these forces are sensed by that cells and they process it or convert it to that chemical signals and do all the jobs. And to understand all the phenomenon, how that fluidics platform is essential to mimic that biological means confinement or biological reactions by which we can understand that in vivo biological phenomenon more or less in details. So, what we learned from this lecture, what extent the stimuli of biological respond to, in what scenario inside in the body mechanical factors come into play, then what are the mechanosensitive or mechanoresponsive components of the cell, how cells integrate mechanical or biochemical networks, and how can biomicrofluidics help to study such phenomenon. Now, if you see that cell that is in its units of all living system is a viscoelastic material if we look for that physics point of view, and it is surrounded by that extracellular matrix protein, like say these are the two cells and these are the surrounded by the extracellular matrix proteins. This extracellular matrix proteins provide the surface or provide that support to sustain that cells, and these ECMs extracellular matrix, it contains mainly that protein collagen and elastin. Collagen provides the tensile strength, stiffness, as well as it hinders that your strain due to compaction, that cyclic strain by which that cell can sustain. So, now we can understand that cell is a viscoelastic material and extracellular matrix also has a mechanical properties. So, these cells and that extracellular matrix, they give the signal to each other by which they can manifest their programs, or you can tell that outcomes. So, if you can see that here that cells, it is acting its own mechanical properties and ECM architecture at the same time chemical signals, means cell can integrate all the physical forces together, then convert to that is your chemical signals and do all that biological outcomes. Now, what are the chemical signals? Chemical signals are growth factors, hormones, neurotransmitter and etcetera, and mechanical stimuli are like say pressure, touch, movement of sound waves and etc. If we look for that extracellular matrix point of view, so they give the signal from outside to in, means that stiffness of that extracellular matrix, it provides that your fluid stress, then tensional force, then compaction force. And on the contrary, cell will provide from their chemical signal process to mechanical signal through focal adhesion site gives the transmit the force. So, this is signal inside to out. So, signal outside in and inside out, they feedback each other and do the biological outcomes. So, look for that what are the means your chemical signals. In our biological fields, we know that all the chemical reactions are governed by your reaction diffusion. So, here that chemical signals are recognized by that cell surface receptors, basically they act as a antenna like things. So, these receptors will recognize that outside chemical signals, they transmit that signals to second messenger molecules, then so on and so forth, ultimately go to the nucleus and they process that message. So, if you look for that this diagram, here that ion channel receptors are there in presence of particular molecules acetylcholine, that ion channels is opened up, then it opened the channel for passing that all the ions from outside to in. So, if you can classify that receptors are of three types basically, one is your membrane receptors class, another is G protein coupled receptors and ion channel receptors. So, just now we discussed ion channel receptors. And what are the G protein coupled receptors? There are varieties of signals like say light, ions and your different types of hormones and etc. They bind to their specific receptors, like say these are the specific receptor, then they pass the signal to G protein. Now, the G protein will trigger that membrane bound some enzymes or say ion channels to give the second messenger molecules. Like say if you take an example of say adrenaline, it is a stimuli, that adrenal receptor in that membrane, it acts as a receptor, then G protein acts as a transducer. Cyclic AMP produced from that adenine cyclase, that is amplified signal, then this amplified signal activates the protein kinase A, that is acts as a sensor, and it is then it is going to the nucleus, then effector molecule is transcription regulator, then gene is activated. Means outside signal is recognized by the receptors, then they pass that signals and these signals are diffused in the cytoplasm, then they go to the nucleus, then they give the in the form of messenger RNA transcription. Whole the process it takes around 5 seconds to 10 seconds. So, if we summarize all that your chemical signal transduction pathways, basically there are around say several pathways and near about 16 or 17 pathways, and there are some pathways which are signals are generated inside that cytoplasm. These metabolites and other signals work by that either protein protein interactions or second messenger molecules, and these signaling pathways more or less they always cross talk with each other and for specific biological outcome. On the other hand, if we look for that in our body, what mechanical stimuli we face? We face different type of mechanical stimuli in the different parts of the body, say shear stress, interstitial flow, transendothelial flow, stretching. These are, if we go through that diagram, actually we can see that in vasculature we can see that shear flow, interstitial flow. In the muscle we can see that stretch, active tension. In case of neural tissue we can say that ECM stiffness. Then epithelial tissue we can look for the stretch, shear flow, and a connective tissue cyclic strain and interstitial flow and compaction. Moreover, that ECM extracellular matrix which provides the stiffness of the tissues, it has also a architecture like say topography in the nanoscale topography, which is very much important for functioning the cells. And from this diagram we have a little bit idea what types of mechanical forces are imparted by that fluid flow or compaction. The decision means how mechanical inputs like say shear stress, interstitial flow is going to the tissues, then tissue with its intrinsic mechanical properties like say stiffness, viscosity, it is converted to biological outputs like say your altered gene expression, protein secretion, cell migration, differentiation. And then this goes to the feedback to mechanical input by which tissue can modulate itself to do that biological output. Now, how these two signals means mechanical signal on the other hand chemical signal differs basically. So, biochemical signals or chemical signals, these are of diffusive in nature, whereas mechanical or physical your force, it is a displacement like nature, it is a basically it makes a deformation of the cells. Otherwise, that transmit that force by cell cell interactions or through extracellular matrix proteins. Next is that chemical signals are transported or transmitted by your simply by diffusions, that is why it takes a long time. Whereas, mechanical signals they transmit by their compaction. So, there is not much loss during the transmission, maybe due to some damping, but that not too much basically. That is why your mechanical signal transmits within a say 5 microseconds, whereas chemical signal transmits around say 5 to 10 seconds. And last difference is that cells sense that your chemical signal in a 2-dimensional way, like say chemotaxis, it is 2-dimensional and it sense that your chemical signal 2-dimensional, whereas in the mechanical signal they sense in totally 3-dimensional way, that is that 3D force they sense. So, we can have an idea how that mechanical signal differs from biochemical signals, but at certain point of time that mechanical signal will be converted to chemical signal. That is why that mechanotransductions is coming to that scenario. So, essence of biomicrofluidics should study the mechanobiological responses are that to closely mimic that cell's natural microenvironment means which environment or which confinement cells exist along with that mechanical forces, and to create a continuous perfusion system and chemical gradients, or you can tell that mechanical cues gradient, to apply fluid shear or different flow patterns to study the cellular behavior. Like say in our body we have pulsatile flow, but in all the cases pulsatile flow may not exist basically, particularly in the venous situation it is a steady flow. So, we have to create different types of flow to study that cell behavior, what is occurring in vivo, if we can mimic that and in vitro to simulate that real situation. So, what are the mechanosensors in biological cells? First one is your stretch activated ion channels, means when membrane are stretched, these ion channels are activated. In endothelial cells, the glycocalyx, there is a glycoprotein like things which are protruded from that cell surface. These are also acts as a mechanosensor, because on flow these moves basically. Then cell junctional proteins, this is adherent complex, and cell ECM, that junctional complex, that focal adhesion complex basically. Your force induced unfolding ECM proteins like say fibronectins, which are their active sites or some peptide sequence are cryptic, but on force those cryptic sequence are exposed, and by which integrins of that cell surface protein they can interact. Then intracellular strain can induce conformational changes in cytoskeleton, by which that motor proteins and other assembling structures are changing and mechanical signals are produced. And that nucleus itself acts as a mechanosensor, because that mechanical force directly may transmit through actin, myosin or tubulin fiber to nuclear envelope, and then they can activate the chromatids. That compression of intracellular space can alter the effective concentration of the autocrine or paracrine molecules, that is means your effective concentration at that confined space is increasing also. Besides this, there are others, a lot of G protein coupled receptors, lipid fluidity also involved during that your fluids here. So, two important mechanosensors are say focal adhesion complex and your lipid rafts are very much important in our study. So, I shall discuss a little bit about that focal adhesion complex. Focal adhesion complex, it is a acts like a clutch in a mechanical system, which engage and disengage between the two systems. Here that focal adhesion acts to engaged cell to the ECM and again disengaged cell to ECM and transmit the force accordingly. That focal adhesion complex basically it is a very dynamic in nature. It is starts initiation, then growing, then mature, then dissociates. The whole process takes around say maybe 20 seconds or less according to the situation, and it starts that when that actin filaments are means polymerized, it gives the stress or give the force to that cells and then ECM. This process triggers that actomyosin complex to form that molecular or compaction force, then it recruits lot of modules, this is adapter proteins, then signaling molecules, then actin polymerizing module. And whole this complex again triggered that actomyosin complex may compact and give the force, and this your signaling molecule triggers that small G protein or the GTPase etc. Then they triggers that cytoskeleton regulating proteins, then force generation. In that process it is a recirculating, and that focal adhesion is growing around say 3 micrometer area, and it imparts force or transmit the signals from ECM to cells, and topography of ECM and that stiffness of ECM influence that focal adhesion strength. So, this is you can tell that outside in signal is provided by that focal adhesion complex, and when focal adhesion dissociates, when that actomyosin complex, that contraction force is much more than that focal adhesion force, around say 5 micronewton, 5 piconewton per micrometer squares. So, at that situation focal adhesion dissociates and again it starts focal adhesion point in other place of the cytoplasm. In that way focal adhesion transmit that mechanical signal inside the cell and it converts to chemical signals. What is lipid raft? Lipid raft is a micro domain in the cell membrane. It is also very dynamic in nature. Its dimension around 1 nanometer to 200 nanometer. It is enriched in cholesterol and sphingolipids. It is regulates activated the lot of mechanosensitive plasma proteins, basically plasma proteins, or you can take that protein complex in addition to focal adhesion proteins. And it acts as a platform for cytoskeleton protein linking ECM and organizing cellular trafficking, means that lipid raft can be transmitted from that cell surface to Golgi, then again it is coming to the cell surface even in going to the focal adhesion also. So, that is why it is very much dynamic and its size ranges from 1 nanometer to 200 nanometer dimension, and its size and that aggregation depends upon that fluidity of the membranes, membrane shears and etc. So, as I told you that mechanical force directly go to the nucleus by the help of your actin filaments, nespirin, then it is going to that lamine, then it is you are acting to the chromatin to transcribe its all the activities, or you can tell that messenger RNA, tRNA synthesis. So, what you can tell that mechanical force from outside going through focal adhesion complex or other mechanosensors through actomycin complex or microtubulin, then this is directly in contact with that nuclear envelope proteins and it is transmitted to chromatid for their transcription and your other activities. In our body, each and every tissues and cells in the tissues faces that mechanical force. So, this is an example how that in endothelial cells which faces that blood flow, it faces that means your mechanical force and it converts to chemical signals. So, in the as I already mentioned that in endothelial cells they have that glycocalyx, and these glycocalyx are protruded from the cell surface when fluid flow during that stress is developed, and under the stress this is bending basically, and this triggers that other ATP release activity, then stress activated ion channels, and that mechanical force is transmitted to that cell junction and focal adhesion points, and it starts that endothelial activity. And in endothelial cells should be properly connected for their proper functioning, and that is very much essential for its integrity. And fluid flow means your blood flow is means types of blood flow, whether it is a streamline or turbulent, influence that activity. In cardiac muscles, that is cells are cardiomyocytes, and it is a mechanical force acting organ, it generates that force. You are seeing that is heart beating basically, means each and every cell of the myocyte, cardiomyocyte beats, and that beats starts with that lot of integration of that mechanosensitive sensors like say stress activated ion channels, G protein and intrigants, and they convert these mechanical force to chemical signals through RAS, NOS, nitric oxide, then they convert to nucleus like NFAT, NF-kappa B and etc. So, cardiac mechanotransduction signal is mainly that mechanical force converged to chemical signals inside the cells and do its beating job. The tumor is a nice example where chemical signals and mechanical signals integrated together and they provide that signal ins out, outside in signals. There are few examples I am citing here, where that microfluidic platform is essential to understand that physiological behavior, like say micro confinement and that your mechanical force, fluid stress, etc. This is an example. Say we know that atherosclerosis plaque in heart disease basically, and plaque develops where that bifurcation is there. Where that your blood flows means your parallelly to that channel, means your without any turbulence, particularly this portion, there is no atherosclerosis plaque development. Whereas, when the turbulent flow is there, that atherosclerosis plaque is developed due to inflammation, leakiness, lot of things are there. Now, question is why this is happening? To answer this question, microfluidic platform is the answer, because when your endothelial cells are growing in the microfluidic channel, when you are giving the flow around 0.07 to 13 Pascal, then the cells are aligned properly in the direction of fluid flow. But if we give a less flow or in turbulent flow, then cells are not arranged properly, cells are not adhered properly, and that means leaky. So, possibly this is the answer, that is from that basic fluid mechanics we know that where that bifurcation is there and that where that channel diameter is higher, then fluids flow becomes turbulent. So, in the turbulent flow that cells are leaky. So, that is a very interesting observation, we can prove what is happening in the nature in the microfluidic platform. This is another example. Without microfluidic platform, we cannot understand that physiological phenomenon in that renal tubular cells, epithelial cells, where it means by osmosis or hydrostatic pressure, they pass that sodium chloride and activates that renal filtration process. If we culture that renal tubular epithelial cells in normal glass plate, then polarization we cannot see too much. Polarization means one is your basal layer, another is apical surface, and both the surface has a different functions, they express different proteins. But if we used some your multilayer microfluidic platform, then the cell behaves or polarity is nicely visible, and you can see that here, that apical surface which faces that hypertonic sodium chloride solutions, they express that aquaporin, whereas in the basal layer which is faces the interstitial flow, that is express that sodium potassium pump, and at the same time you look for that height of the cells is normal in the microfluidic platform, whereas normal glass surface it is in the flattened. So, microfluidic platform it gives that cellular niche mimicking that physiological system. In that way there are huge amount of examples by which we can understand that your physiology by using that microfluidic. There are examples like say shear stress, if you give erythrocyte for 4 to 3.3 Pascals ATP release. In the bone, if you give 3.4 Pascals osteoblast differentiation, shear magnitude depend on the calcium influx and osteoblast. In that way there so and so forth, lot of examples are there. Here I am showing only few examples like say confinement of the cancer cell, rapid directional persistent migration, then compression the neurons at 65 Pascal, focal axonal swelling, altered axonal transport, stiffness in neurons that with this 57 to 797 Pascals axon outgrowth. In that way lot of examples are there. So far in the microfluidics area, we are introducing one or two variables to understand the physiology. So, next fluidics is coming to that how we can integrate different types of force together to understand that what is happening in real biological system, along with stiffness of the substrate, your fine topography or nanoscale topography of the surface, then shear stress, interstitial flow, then compression force, everything should be integrated in one platform, by which we can understand that physiology of the particular cells and particular biological environment. I think I can impart some idea about that why biomicrofluidics is necessary to understand that physiological phenomenon, and if we can mimic this system in fluidic system, we can develop some disease model, and at the same time we can observe some biological phenomenon so far not possible to observe in the traditional system. So, that is why that microfluidics is important to understand that biology. Thank you.